From the JAMA Network, this is JAMA Ophthalmology Author Interviews. Conversations with authors exploring the latest clinical research, reviews, and opinions featured in JAMA Ophthalmology.
Hello, and welcome to this author interview from the JAMA Network. I'm Neil Bressler, Editor-in-Chief of JAMA Ophthalmology, and today we're going to look at an investigation on the assessment of Parafoveal Diabetic Macular Ischemia on OCT angiography images to try to predict diabetic retinal disease progression and visual acuity deterioration. And joining me is the corresponding author for that investigation, Dr. Carol Cheung. Dr. Cheung is an associate professor with the Chinese University in Hong Kong. Carol, thank you so much for sharing your work with us.
Thank you, Neil, for inviting me in this podcast.
Maybe we could just start with what your group meant by diabetic macular ischemia on OCT angiography. What was the definition of that?
Yeah, thank you for the question. So actually, the DMI defined in this study was based on OCTA images with reference to the early treatment diabetic retinal study protocol based on dye-based angiography. So we know that our OCT-based DMI definition here cannot directly compare with the ETDRS based on dye-based angiography. So in fact, we hope that this can be an open area that experts more people to discuss what we can have a more accurate term for DMI OCTA to distinguish DMI identified by fluorescent angiography would be preferable.
Now, I understand that typically we could look at a fluorescein angiogram and see areas where perhaps the flow appeared to be missing on the fluorescein. Is that what you're looking for on the OCT angiography as well? You're looking for areas that detect flow where perhaps they're missing around the foveal avascular zone?
Yes, we want to look at the non-perfusion area. In fact, I don't think OCTA can look at the flow because it's still a structural images. The OCTA is based on the flow concept to generate the superficial capillary plexus or other capillary network. But this is not a flow signal. But what we give is the structure of the capillary network. What is the ischemia area that we can quantify?
I see. So you're looking for these areas where the structure of the capillaries might be missing. Now, you mentioned that there's superficial capillaries. Could you just describe to us, I think there were 3 areas that you looked for capillaries, different layers that you look for. Could you help define those for us as well before we get into what you actually studied in those areas?
In the retina, actually, right now, the techniques can distinguish 3 capillary network. One is superficial capillary plexus, intercapillary plexus, and also deep capillary plexus. In our study, we only look into superficial capillary plexus and deep capillary plexus because the OCTA algorithm we use is not able to identify the interlayer plexus. So we more focus on this superficial capillary plexus and deep capillary plexus in our study. And on each capillary plexus, the OCTA image, we use a deep learning algorithm that we developed before that to identify presence or absence of DMI.
Now I'm beginning to understand it a little more. So you're looking for these capillary structures in the macula, and you're going to look specifically on OCT angiography at these capillary structures superficially and deep. You're looking at absence of these capillary structures. Do we know if that really represents ischemia or infarction? I know you called it ischemia. Is it possible that those areas might not be starving for blood flow? They may actually be areas of infarcted tissue.
This is a very good question. In fact, we don't know. I think, still not sure that how to distinguish that because it is not a dynamic imaging analysis. So what we look at is really looking at missing capillaries. Then we measure the area. And in this study, we actually do a binary classification whether there's yes, no, diabetic macular ischemia. But I think the question you mentioned, we need to further explore and further investigate.
We'll call it DMI or diabetic macular ischemia for now, but I understand these are areas where the capillary structure are missing, and they could represent DMI meaning ischemia or DMI meaning an area of infarction. Okay, so you're going to look at these areas and answer from your analysis of these images, yes or no, to try to predict, will the retinal disease progress and will there be visual acuity deterioration? By progression of retinal disease, do you mean the diabetic retinopathy severity scale level? Do you mean diabetic macular edema? Do you mean both?
We mean both. First, we look at diabetic retinopathy progression. So we base on retinal photographs from baseline throughout the follow-up using a modified Early-House classification system. So DR progression here was defined as an increase in 2 or more steps of severity level compared with baseline. So for DME development, we look at OCT. So the development of DME was defined as occurrence of either retinal thickening. We use the definition according to DRCR.net protocol to use the vessels to define retinal thickening. And also we look at the presence of DME features to define DME, such as intraretinal systolic space, etc. Yeah.
Very good. So let's get into a little bit of how you set up the methods for the study. You looked at over 300 eyes. How did you select the patients that were going to be analyzed in this study? Were they prospectively identified and enrolled? Were these part of another study that you had? How did you actually find these 178 patients?
So we have a longitudinal cohort in Hong Kong. So we have recruited a group of patients with diabetes. Doing all the imaging regularly, we follow up in our CHK eye center. So the inclusion criteria in the beginning is if they have diabetes, and then that meet our criteria. For example, they didn't do any surgery, or they don't have other AMD, other major retinal disease, then we recruit them. But specified to this study, we only include study subjects with mild, moderate DR and no DME at baseline.
All right, so we have these eyes that are either mild or moderate non-proliferative diabetic retinopathy. You look at the OCTA in either the superficial or the deep layer or both, and you grade it as yes or no, and then you look for progression. What'd you find? Tell us first in terms of diabetic retinopathy severity scale progression, or this modified Early-House classification that you used.
Yes. So we find that presence of superficial capillary plexus DMI is associated with DR progression. And we also find that presence of deep capillary plexus DMI also associated with DR progression. After adjusting for established risk factors, for example, age, duration of diabetes, fasting glucose, blood pressure, baseline DR severity, ganglion cell, inner plexiform layer, axial length, etc.
And how about the superficial versus the deep? Was one more predictive than the other, or they just were both predictive and independent of one another?
Yes, they are both independent one another.
And tell me a little bit about whether this having the DMI ischemia or not having the DMI ischemia in either of these areas, how is that related to progression or development of diabetic macular edema? Let's just say the thickness of the central subfield on the OCT at follow-up.
Yes. So what we find is that only that deep capillary plexus DMI is associated with DME development. So we did not find superficial capillary plexus DMI associated with DME development. So only deep capillary plexus DMI is found.
If we're able to perhaps have this analysis of OCT angiography at many of our clinics in the future, would we be able to use this instead of having to grade the retinal vessels as mild, moderate, or severe non-proliferative retinopathy? I know that's often hard for people to be certain that they've identified areas of intraretinal microvascular abnormalities, or IRMA, or to be certain they've identified venous beading. Might we use this instead for diabetic retinal disease progression, or would we use this as an adjunctive or complementary measurement to what we already would be grading the retinal vessels for?
I will say that this can be a supplementary tool because a lot of signs cannot be replaced by current retinal photographs. For example, exudates, cottonwood spots, and also retinal hemorrhage that we cannot find from OCTA. So I will say that this is a supplementary, more target to DMI. I would say that this is the first step to identify presence of DMI, and then we can use the quantitative OCTA metrics that what people widely use, for example, foveal vascular zone area, vessel density, etc., to further estimate the extent of DMI.
You looked at yes, no, is there this diabetic macular ischemia? Do you plan on looking at that as a continuum where there's more or less ischemia in the future to see if that identifies more or less progression?
Yes, that's what we want to do. So I think the direction may be, for example, like if persons have presence of DMI, then we can further quantify the extent of the DMI, how serious is it? Because previous studies already showing that, for example, lower vessel density and also larger foveal vascular zone area is related to DR severity or DME. So in fact, I think that if no DMI from OCT, they can more focus on other measurements to predict other diseases.
Do you think we can generalize this to other patients with diabetes, either in your clinic or elsewhere in Asia or throughout the world, or we're not ready to generalize these findings yet?
So this is in fact a first study, but what next step we want to further verify, replicate this association. And also that in this study, we only use one type of OCTA device. We also want to see other OCTA devices can have similar findings.
And should this change our practice yet? If somebody has an OCT angiogram that suggests that there's some ischemia around the foveal vascular zone, or maybe they have a fluorescein angiogram that they obtained and they see that, should they think of that as a patient who may have a higher risk of progressing in terms of their diabetic retinopathy or progressing in terms of developing diabetic macular edema, or is it not yet ready for prime time in the clinical practice?
I think because the dye-based studies have already shown that if there's a diabetic ischemia from fluorescent angiography, studies have supported that they are at risk of DR progression. So I think it's more or less ready, but I will say that more validation, more verification will be tested to verify the association before we can use in clinics. And particularly, I think why this field is hot is there's ongoing clinical trials to highlight the need to better define how DME fits into the DR severity scale, as well as there is a potential new therapeutic target for arresting DR progression and VA deterioration. So we hope that this can support the work on those clinical trials, how to define DMI.
Finally, what are the other next steps that your group might be planning, or others, in terms of facilitating the ability to identify this? As you said, it predicts DME progression in the deeper layers, but not in the superficial layers. So how can we study this further, or what are the next steps that you might do to figure out ways of refining this?
Yes. First, in this study, we only focusing on 3 millimeter by 3 millimeter images for assessing the parafoveal DMI. So we know that actually the larger area, the wide field area is important. But we also facing the resolution trade-off issue between field of view and image quality. So we are working on some other techniques to enhance larger area images to make sure that we can clearly identify the capillary network that we, in order for us to define present or absent of DMI. So this is one of the direction we are working on. Our algorithm was developed based on one OCTA device. We have tried another brand of our commercial OCTA device. And now there's several new OCTA devices. So we hope that our algorithm can generalize in other OCTA devices as well. So lastly, of course, we need clinical data to further replicate the association. We hope that there's more evidence to support that this DMI classification can fit into our diabetic retinal disease staging system.
Well, Carol, I want to thank you and your colleagues so much for helping everyone in the world to try to improve our management of diabetic retinopathy and better predict who may be at risk of developing these complications, not only in Hong Kong, but around the world. So thank you so much for joining us today.
Thank you, Neil, for the invitation.
I'm Neil Bressler, and I've been speaking with Dr. Carol Cheung, Associate Professor at the Chinese University in Hong Kong, about an assessment of Parafoveal Diabetic Macular Ischemia on OCT angiography and how it may be used to predict the progression of retinal disease in people with diabetic retinopathy and even predict visual acuity deterioration. You can find a link to the paper in this episode's description. Thank you for listening to this JAMA Ophthalmology podcast, and to follow this and other JAMA Network podcasts, please visit us online at jamanetworkaudio.com. This episode was produced by Daniel Morrow at the JAMA Network. Thank you all for listening.